Hi everyone. Today I'll be presenting an article titled Precision Tumor Recognition by T Cells with Combinatorial Antigen-Sensing Circuits. So this article was published in 2016 by Owoeye and a list of other authors on the screen. It was published under the journal Cell. This video abstract is presented by myself, Owoeye Oluwamayowa Esther, a postgraduate student of Macquarie University, Sydney, Australia. So, to start right at the beginning, what is immunotherapy? Immunotherapy is a treatment that uses certain parts of a person's immune system to fight cancer. Immunotherapies are thought to work by slowing the growth and the spread of cancer cells, and this helps the immune system destroy existing cancer cells. The recent advances in immunotherapy have revealed that T cells can be used to precisely recognize or identify tumor in the body. So, what exactly are T cells? T cells are also known as T lymphocytes, and they are a type of white blood cells that is of key importance to the immune system. T cells play an important role in adaptive immunity system that tailors the body's immune response to specific pathogens. The T cells are similar to warriors who search out and destroy targeted invaders. So how do we use T cells in immunotherapy? Two major ways. Firstly, they rechannel T cells to recognize and eliminate tumors using chimeric antigen receptors, otherwise known as CARs. The second way is engineering T cell receptor TCRs that combine tumor specific antigen. T cells have also been used to target B cells malignancies. This was done by using CARs directed T cells to the B cell specific antigen CD19. This is the most successful T cell therapy reported. So the use of T cells is actually a very limited approach. This is because it is constrained by the scarcity of tumor-specific single antigens. What does this mean? It means that it is restricted due to the scarcity of single highly specific tumor-only antigens in the body. And this is an important problem because when T cells target antigens also find on bystander tissues, so these are tissues that don't have any tumor on them, it can result into life-threatening adverse effects. The best approach to enhance target-specific activity of therapeutic cells is to engineer them to require combinatorial antigens. Previous strategies by scientists to develop multi-input control of T cells are focused on two chimeric antigen receptors in the same cell, each with partial signaling functions and unique extracellular antigen recognition sites. This approach, however, relies on delicately balancing the same set of coordinated signaling events that occur downstream from the chimeric antigen receptors. To construct more reliable multi-antigen response, it would be ideal to have new receptors that function independently from the chimeric antigen receptor and T cell receptor pathway, but can interface or interact with the chimeric antigen receptor activity in a controlled manner. So this hypothesis led to the aim of this research. In this paper, these scientists showed that it is possible to construct combinatorial antigen recognition T cell circuits in which a CNOT receptor for one antigen drives the expression of a chimeric antigen receptor CAR for a second antigen. This slide here is an extension of the experimental design. It illustrates the combinatorial antigen recognition T cell circuit. The first experimentation method done in this research was to test for CNotch-gated CAR expression in JUCAT cell. The first attempt was to engineer combinatorial antigen control over the activation of JUCAT cell. The authors observed that JUCAT T-cell activation was only observable for tumor cells that express both CD19 and mesothelin, and not tumor cells expressing either single antigen. So, given the success of this test, the next test done was to know whether the same type of CNotch-driven CAR expression circuit could function in In the test to know whether the same type of CNOT-driven CAR expression circuit could function in primary T cells to discriminate multiple antigens, the authors tested various CNOT receptors in primary T cells and revealed that the Gal4 VP64 transcriptional activation domain worked reliably, yielding good CNOT receptor expression and minimal basal transcriptional activity. They also observed that T cells only showed activation when exposed to target cells expressing both GFP and CD19 on their surface. This was revealed by cytokine production. They also tested whether T cells could be targeted to tumors in vivo via CNOT receptors and only express the CAR when in the tumor microenvironment. To perform this experiment, bilateral xenograft CD19 plus Dowdy B cell lymphoblast tumors were used, and these were injected into immunocompromised mice. Luciferase expression was monitored as a reporter for CAR expression over the course of 11 days. The authors observed that the T cells started to express the CAR selectively in the GFP plus Dowdy tumor by day 1 and continually increased local expression of the CAR over the 11 days period in the dual antigen tumor. Finally, these findings show that T cells engineered with dual receptor circuits recognize combinations of antigen and can effectively kill target tumor cells in vivo without harming bystander cells. This is because these dual receptor and gate T cells are only armed and activated in the presence of dual antigen tumor cells. This type of precision dual receptor circuits paves the way to immune recognition of a wide range of tumor in various organisms. Thanks for listening.